When perils arrive on the farm, America's agriculture producers deserve fair compensation that is delivered timely and with confidence. To accomplish this, crop insurance adjusters need loss adjustment procedures that are understandable and accurate. These procedures are developed from original research conducted by the National Crop Insurance Service's Agriculture and Technology Research Program. The program has been conducting crop loss research since 1923. Then and today, researchers have strived to understand the effects of perils on crop growth and yield. The research is very unique in that few other people in the world intentionally damage crops that they are researching. Conducting research requires much deliberation. The first step is to discuss where to conduct the research and who should conduct the research. Once the location and researchers have been identified, a protocol for the research is developed and the project is implemented. Deciding the location for research projects depends on several factors. Of course, we want to conduct research in areas where the crop is grown, but more importantly, we want to place the projects where we can capture the variability in crop response that may be inherent across latitudinal and longitudinal gradients. For field corn research, most of our locations are scattered across the Corn Belt, as this is where the vast majority of corn acres are. This gives us a damage response from corn of differing relative maturities and in differing climatic conditions. For soybean, a similar approach is taken. The locations of North Dakota, Iowa, and Indiana are perfect for testing multiple maturity groups from the low 1s up to and above 4.0s across differing climate zones. The soybean in these areas are predominantly indeterminate varieties. If we want to determine how determinate varieties respond, we have to go south of southern Missouri, and where better than the soybean growing region of Arkansas. However, for some crops like sorghum, we are limited in locations, as the sorghum growing region has limited spread east to west and is predominantly in the central and southern Great Plains. Kansas and Texas are the top two states for sorghum acreage, and these locations provide a large difference in climatic conditions and maturities used. The next step in conducting research is to identify who will conduct the research. Because NCIS has conducted crop loss research for 95 years, we have developed a network comprised of the best field-based research experts at leading land-grant universities. They are typically cooperative extension researchers that have a mission of delivering results of field-based research to the public. They are also specialists in the agronomic management of specific crops and have the available resources including labor, equipment, and available plot space at the research centers. They are our independent, unbiased third-party partners that allow the crop insurance industry to maintain an arm's length from the research that drives crop loss adjustment procedures. Prior to conducting any research, a protocol is developed. It is a standard operating procedure for the project that is much like a roadmap that guides the researchers over time. This single page was the advent of the protocol for the recently completed Green Snap project that was developed from an initial meeting in the fall of 2014. A protocol takes from several weeks to several months to develop as ideas are tossed back and forth among the researchers and NCIS. In this case, the single page blossomed into a full set of operating procedures that all of the researchers adhere to. Let's take a closer look at this protocol and see what it entails. First, the researchers involved and their affiliate institution is listed. Next, the guidance for any protocol development is the research question or questions that we want answered. It keeps the researchers focused on our intentions for the research and provides a quick reference to other people working on the project. In this case, of the Green Snap project, the research questions arose from discussions in a Crop Hail PPLA committee meeting. Using the research question as guidance, we were able to tap into the expertise of the researchers and develop an approach to conduct the research. This section describes what researchers call the plot treatments and any additional explanation of the treatments if needed for future reference. The treatments explained in the approach are always listed in table form for quick reference by anyone working on the project. Here we see that each treatment has a number for cross-reference, and that each treatment is a combination of the growth stage, or when the treatment will be applied, the percent of plants snapped, or what is to be applied, and where the snap is to occur. Next, we see that the number of replications is specified, as well as the plot size and how the treatments are to be applied to the plots. We will discuss these items shortly when we go to the field to implement the project. All protocols mention agronomics or cultural management that refers to standard grower practices for the area in which the plots are located. The last consideration in this protocol refers to the data collection. For this project, the data collection refers to what is to be sampled, how it is to be sampled, and what other data is going to need to be collected. Now, it is time to get the project in the ground. So let's go take a closer look at what the protocol entails as we implement the project. While many projects are simply carved out of bulk planted fields of the same hybrid or variety sown at one seeding rate, such as that for the corn green snap project, others are more complicated, such as when 2 or more plant populations are used, or when plots are split to accommodate in-season subsampling or extra treatments. Irrespective, soon after the crop is planted, the location of the individual plots are determined and a plot map is finalized. To explain this process, let's look at the treatment table in the Green Snap Project protocol. Here we have our table of 13 treatments. As the plots for the 13 treatments are laid out in the field, the plot map would look like this and constitute one replication of all 13 treatments. One replication does not provide much information because there is simply one observation for each treatment. So to get more accurate data, treatments are replicated multiple times. However, each treatment is randomly assigned to a different plot in each replication in order to make a statistically valid experimental design. Thus, the location of any treatment in any replication is independent of the location of a different treatment. Keeping the treatments independent is necessary to satisfy one of the assumptions of the statistical model. In researcher language, this common agriculture research experimental design is called a randomized complete block design replicated 3 times. The experimental design of most NCIS-sponsored research is replicated at least 4 times, and sometimes more replications are necessary. You will notice that treatment number 1 is what we call our untreated check or control plot. When the data are analyzed, the yield response from any treatment in any rep is then compared to the untreated control within the same replication. While research projects are replicated in space at each location in each year, they are also replicated in time, with most research projects typically being conducted for 3 years at each location. This allows the data to encompass the variability in growth response that occurs from year to year. Now, let's take a look at how big research plots are. The size of a research plot actually depends upon the crop, the data to be taken, and the treatments applied. In the case of the Green Snap project, the researchers agreed that the plot size is flexible, but that at least 40 ears from undamaged plants would need to be harvested from the 2 middle rows of each plot. If 75% Green Snap is a treatment with the least undamaged plants, we would need 160 corn plants in the middle 2 rows, with 120 of these plants being green snapped. To accommodate this, each plot would need to be about 40 feet long. For our grain sorghum stand reduction research, plots were enlarged to 55 feet by 15 feet to accommodate a treatment with 90% stand reduction. Once the crop in the field plots is up and growing, researchers are focused on tracking plant development and identifying the growth stages so that they know when to apply their treatments. Once the crop has reached the growth stage for a particular treatment, the researchers enter the field to apply the treatment to the plants in the plots. For a better discussion of this, let's turn it over to Dr. Roger Elmore as he tells us how he applied the Green Snap treatments.
What we do, this is a 3 out of 4 break at node 12. We established earlier that node 6 is right at the soil surface, so this is 7, 8, 9, 10, 11, and 12. So this is the 12th node. What I'm doing 12th node breaks, basically hold it here, and then I break away from this axle here, so it breaks like that. Once I establish that node, I really don't look at it again. I just do approximately the same node. So there I'm breaking away from the leaf axle, breaking 3, skipping 1, and just continuing on down the row like that. It's best to do this in the early morning when the dew is still on and the temperatures are cool. Once it warms up and the dew burns off, the plants get a lot more flexible and it's a lot harder to break them.
Roger was applying Green Snap to 75% of the plants in the plot by snapping the plants in a consistent pattern of every 3 out of 4 plants down the row like seen here. However, When perils impact a standing crop, the impacted plants obviously are not consistently damaged as that's shown here. It might look like this, or like this, or like this. These other non-consistent patterns are termed skippy, and for these 12 plants there are a multitude of skippy patterns that could be implemented on the plants. NCIS typically applies damage in a consistent pattern because research has shown that there's not a great difference in the plot response between a skippy versus consistent pattern, and because there are simply too many combinations of skippy patterns to apply. Let's take a closer look at a plot. It is standard procedure in agriculture research to apply the treatment to every plant in the plot and use the middle of the plot for taking samples. In this case, rows 1 and 4 of each plot act as a buffer between the rows we want to sample and the different treatments that are applied to the adjacent plots. And here is a look at the soybean defoliation study and the grain sorghum stand reduction study. For both of these studies, more than 4 rows were planted per plot. Yet in each one of them, the middle of the plot is still the sampling area. The final step listed in the protocol is data collection. Some NCIS research projects are hand harvested, such as when samples are taken during the growing season. However, most projects are machine harvested at the end of the growing season with a specialized research plot combine. This research combine is set up with a 3-row header and is not used for corn, but is used for grain sorghum where plots are set up as either 5 or 7 rows with the middle 3 being harvested. The operator will stop between plots while the combine cycles through the grain and then proceed through the next plot. Here is a picture from the cab of a plot combine with a 2-row corn header that is used for harvesting the middle rows of 4-row plots. One final aspect of conducting research is visiting with the researchers in the field to get a first-hand interpretation of what they are seeing during the implementation of the project.
This is single seed, no cluster. But these two, I think
So we'd be in that, I'll say mid R5 stage yet. And you know, I think partly what is helping these soybeans hold on a little bit is for this year anyhow, we've cooled off.
It is this insight beyond the numbers that helps National Crop Insurance Services develop better research methods for conducting crop loss research, gather better data, and design more insightful research. It is with these principles that the National Crop Insurance Services Agronomic Research Program continually strives to improve the quality of research and the accuracy of crop loss procedures.